Hello biologists, welcome back to Bioscience. This lecture is all about cellular respiration. Let's title our notes Cellular Respiration. Cellular respiration is the process of taking food and getting energy. And let's write that scientific definition there. Cellular respiration is the process of extracting molecules within food to make energy. And that energy that we use in our cells by and large is ATP. We're going to focus on cellular respiration, that breakdown of glucose into energy. Glucose is a simple carbohydrate. That's where a lot of our energy comes from. However, know that you can also do different but similar processes in order to break down proteins and make them into energy, ATP, or similarly lipids, break those down and make ATP out of it, but it gets real complicated. So we're going to just focus on glucose for the purpose of this video. So let's talk about glucose actually, because when we have glucose, and that chemical formula for glucose is C6H12O6, meaning that there's 6 carbon molecules, or 6 carbon atoms, 12 of the hydrogens and 6 of the oxygens. When you combine glucose with oxygen, there's going to be 6 of them actually, 6 oxygen, that result will end up ATP, but we also get some side products here. We get H2O, we get 6 of them, 6 CO2s, and we get 36 to 38 ATPs. Kind of depends on who you ask, but let's stick with 36 to 38 ATPs. So we have water, carbon dioxide. You mostly exhale that carbon dioxide out. I spelled that wrong. Let me fix it. Dioxide. That's better. Carbon dioxide, and then ATP, that's that cellular energy. There's an overview of cellular respiration is 3 major steps. So 3 steps. Those 3 steps are, the first step is glycolysis. Glyco refers to glucose, and lysis means to break, so it's the process of breaking glucose apart. Then there's step 2, that's the citric acid cycle. Also known as the Krebs cycle, named after some guy Krebs. And then the last step, step 3, is the electron transport chain. So let's kind of focus in on each of these steps. Let's start with an overview of where this happens. And when we talk about making energy, well, mitochondria, it's the powerhouse of the cell, right? The mitochondria is what make our energy, and it does this because it has this really special structure, and each of these 3 steps occurs in a very specific place in order to best facilitate how we make that energy. So let's talk about mitochondria. So mitochondria kind of looks like a bubble, like this. And within that bubble of the mitochondria, then there's this inner bit that kind of wobbles like this. So this is mitochondria. So that mitochondria. We have some structures here. We have this outer part is called the outer membrane, made up of the same stuff that plasma membrane is made of, phospholipid bilayer. That's what those membranes are made of. Then we have this part on the inside, that's called the inner membrane, And lastly, there's the space between, so we call that the intermembrane space. So all of this space in between, everything that's in between those two membranes all falls within that intermembrane space. I could keep going, but you get the point, right? So all of this space here is called the intermembrane space. So we have our membranes, we have our intermembrane space, and then we have this stuff in the middle. This is called the mitochondrial matrix, or just the matrix. And remember that a mitochondria is an organelle, and organelles live inside of cells. So that means it lives within kind of the goo inside of that cell. So outside of the mitochondria is the cytosol. Outside. And all of this happens within a cell. And all of these three steps occur somewhere in relation to the mitochondria. So let's chat. So let's see, I'll label glycolysis. I'll label my processes in red. So glycolysis occurs in the cytosol. So one, glycolysis. Glycolysis, that occurs in the cytosol. Let's talk about glycolysis, actually. Let's talk about glycolysis occurring in the cytosol of the cell. So one, glycolysis, taking glucose and breaking it down. So it happens outside of the mitochondria, but within the cell. So it just happens within cytosol. Close parentheses. Okay. Essentially what happens is we take glucose, C6H12O6, and then there's like bunches of steps. There's 10 steps to be exact. 10 steps. Each facilitated by an enzyme. 10 steps. So from glucose, C6H12O6, there's a bunch of steps, and each of those 10 steps has its own enzyme that does that specific step. And your result is this molecule. It's 2 pyruvate molecules. Then we also get 2 ATPs, and then we also get this guy, we get 2 NADH. NADH is a new one that we haven't heard of, and so is pyruvate. So let's break that down. Pyruvate, I'm going to label in blue. Pyruvate is a 3-carbon sugar. So if glucose is a 6-carbon sugar, we're breaking glucose basically in half into 2 3-carbon sugars. We get ATP, that's energy. But we also get NADH. NADH, similarly to ATP, has some energy we can use later. So that NADH, it holds energy we can use later. And we will use it later, that held energy. Okay, so we take 6 carbons from our glucose and we split it in 2. Now we have 2 3-carbon sugars in the form of pyruvate. That gets us, so we go from, let's go back up, from the cytosol, that's where we did our glycolysis. From the cytosol, we're actually going to jump into the mitochondrial matrix for the citric acid cycle. That is step 2. Let's make a new color. Let's do the citric acid cycle in purple. Purple sounds fun. All right, so let's do step 2 is the citric acid cycle. And if we were to draw a citric acid cycle, it kind of looks like this. It's like a series of steps, and it's a cycle, so it kind of goes around and around and around. So that happens in the mitochondrial matrix. All right, so let's talk about the citric acid cycle, also known as the Krebs cycle. So we have glycolysis. Step 2 is the citric acid / Krebs cycle. The citric acid / Krebs cycle happens in the mitochondrial matrix, or just matrix for short. And we take these pyruvates. We take those pyruvates and we do stuff with them. So let's talk about this. We take that pyruvate molecule, a 3-carbon sugar, and then there's a bunch of steps. So this is 10 more steps. 10 steps, each facilitated by an enzyme. So there's 10 steps. Each of those steps has its own enzyme to do that specific part of the steps. And that gives us a bunch of stuff after. So we have 3 carbon dioxide, CO2. We get 2 ATPs. That's great. That's energy. Plus we get 3 NADH. Oh, and I almost forgot a new one, which is FADH2. So we've heard of all of these. We've heard of pyruvate, we've heard of CO2, ATP, NADH, same as before. But this new guy here, FADH2, is kind of the same thing. It holds some energy that we can use later. So I'll label that in blue. Holds energy we can use later. Holds energy we can use later. But, okay, so this was just for 1 pyruvate, for 1. So for 2 pyruvates, this is what the reaction looks like. So let's see, we do times 2 for 2 total pyruvates. So we have now 2 pyruvates. This is the whole reaction, 2 pyruvates. 2 pyruvate. We have 10 steps. Then we get double all of our products. We get double this. So we have 6 CO2 plus 4 ATP, 2 FADH2, and 6 NADHs. Okay. We talk about, and you might look online and notice that it's just talked about as pyruvate instead of 2 pyruvates. That's because each pyruvate molecule goes through the citric acid cycle kind of independently. It goes by itself. And it's commonly drawn like, okay, so this goes here, this goes there, for 10 steps. And it's a cycle, so that's why we call it a cycle. Okay, so from there, this happens in the mitochondrial matrix. Let's go back to our mitochondria. So we started with glycolysis, then we went to the citric acid cycle. Remember that glycolysis resulted in pyruvate. That pyruvate got stuck into the mitochondrial matrix, went through the citric acid cycle, and now we have stuff left over. We have carbon dioxides, we have some ATPs, etc. So where we go next is step 3, the electron transport chain, and that actually occurs in the inner membrane of the mitochondria. So let's choose a color. Let's do green. Okay, so from glycolysis and the cytosol, we'll go to the citric acid cycle within the matrix, but then from the citric acid cycle, then we're going to go to the membrane. That's where step 3 occurs, the electron transport chain. So I'll just label it like this. Step 3, electron transport chain, ETC, within the membrane. So let's blow up a little bit of our membrane. We're going to zoom in. So if I were to take the membrane like this, and I zoomed in on it, we're going to zoom in on that little part, this little part that I just made a square about. Okay, so the last part of our situation here. Okay, so step 3 is the electron transport chain, or ETC, electron transport chain. Okay, basically what happens in the electron transport chain is we start with 6 ATPs. You need a little bit of energy input in order to get going. And then we're going to take 8 NADHs, and we're going to take 2 FADH2s, and then we get lots of ATP. Lots of ATP. The number kind of varies based on who you ask, but like 36 to 38 in total. Okay, so where do we get all of these molecules though? Well, we get them from the earlier steps. Let me scroll back here now that we can see them all on one page. So we have 6 ATPs, and we got those 6 ATPs because we had 4 from citric acid plus the 2 from glycolysis, so 6 ATPs. And then we additionally get those NADHs. We have 2, and then we have 6, that's 8. And then our 2 FADH2s come from citric acid cycle as well. So everything that we're using is from a previous step. And all of that is done in order to generate lots of ATP. So what this looks like, remember we're going to zoom in on a little bit of the membrane, so let's do that. If I draw a little bit of the membrane, boop, boop, boop. Okay, so here's where we're zooming in on the membrane. Remember that membranes are phospholipid bilayers, So that means that we have phospholipid, boop, 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 boop. Okay, so there's all of our phosphate heads, and then we have our fatty acid tails, right? Fatty acid tails. Okay, so in the electron transport chain, we have our plasma membrane, in our electron transport chain, we have our inner membrane. So where we are, that I've gone ahead and drawn, where we are is we have our mitochondria. We have that inner membrane. And this there, so I have my phosphate heads and my fatty acid tails, that's great. But then I've also gone ahead and inserted these proteins, these intermembrane proteins. And these proteins are really, really important for the electron transport chain. Let's label them. They are called proton pumps. So all of these guys are proton pumps. Proton pumps. That's what all these guys are. And another name for a proton is H+, hydrogen ions. That's another name. So, okay, so if I were to zoom in on my mitochondrial membrane as such, from here, that gets me here. So notice that this top part is the intermembrane space, and the bottom part is the mitochondrial matrix. Each of these big old green guys, this guy, and that guy, each of those are protein. They're ion pumps. Those pumps push hydrogen ions across the intermembrane space and the mitochondrial matrix. This last one, this big one here, is ATP. I'll zoom up here. ATP synthase. That's an enzyme that makes ATP. So how does that happen? Let's go back to our reaction. I'll zoom out just a little. Okay, so we have the electron transport chain. We start with some ATPs. We start with NADH, we start with FADH2. The thing about NADH and FADH2 is we can break off some of those hydrogens. So if we break off some of those hydrogens, what this looks like is, so NADH, for example, NADH loses a hydrogen, so then you get NAD plus hydrogen. And then if we do FADH2, we have 2 of them. We have 8 of these, 8 of these, 8 of those. So if we have our FADH2s, then we get FAD plus H plus. So we get 2 of these and 4 of those. So we have a lot of hydrogens, and each of these proton pumps takes one of those hydrogens. One hydrogen kind of goes down, and it goes over, and then it goes through. And so this transport of hydrogen ions is moved such that the ATP synthase can open up its doors and allow for the movement of hydrogen. And when that hydrogen moves or diffuses, that then can lead to the production of ATP. Let me show you a picture of what that looks like. So this is from OpenStax Biology. It's one of those free biology textbooks. I really like this picture because it shows really nicely how these things happen. So NADH and NAD, then we produce that, and that hydrogen that's left gets sent into the intermembrane space. So notice that we get, and then the same thing happens, intermembrane space. There's lots of hydrogens here. And not so many hydrogens here. And remember that diffusion says that when we have a high concentration, we want to move to a low concentration, and that's exactly what ATP synthase does. Takes hydrogen, moves it from a high concentration then to a low concentration, and that process leads to the production of ATP. That process leads to the production of ATP. So let's go back to our own drawing for a sec. So what happens is that we get this high concentration of hydrogen ions in the intermembrane space because it gets moved. And so that hydrogen ions then get diffused through ATP synthase. That process is diffusion. And that allows for this whole thing to work. When ATP synthase diffuses hydrogen into the mitochondrial matrix, this gives enough energy to make ATP. So let me write that down. So when ATP synthase hydrogen ions diffuse. And let's watch this again in our nicer picture. So hydrogen ions diffuse. They go from the intermembrane space from up here down here where they're less concentrated. ATP synthase does that. So H plus, or hydrogen ions or protons, diffuse to mitochondrial matrix. And when they do that, this gives enough energy. Gives enough energy to make energy. Lots of ATP, like 34 to 38, depends on who you ask. So lots and lots of ATP can be done by this process. Again, this is scraping the surface. There are whole classes on cellular respiration. However, this is enough for the purposes of this class and enough to get you started with this week's content. I really hope you ask me if you have any questions, and I hope you have a really awesome week. I've been really enjoying this semester so far, and I hope you are too. Have a good one.